Have you ever wondered what humans are made up of? At the microscopic level, humans are made up of molecules known as DNA. Genes are small portions of that DNA that are inherited by their parents and code for a particular trait. Here's a little history behind genetics. Gregor Mendel, known as the father of genetics, discovered that both parents must pass down discrete physical factors to their offspring. He experimented with garden peas because of their fast growth, contrasting traits, and being easily crossed. After cross-pollination, Mendel's experiment resulted in the finding of how each individual has two factors that determine what external appearance the offspring will have. We know these factors as genes. His discovery, known as the principle of dominance, explains that the offspring's genes will be determined by the parent state of their gene, either dominant or recessive. These instructions are inherited from the parents. Take Erin, for example. She has red hair, which is recessive. Red hair is a recessive gene known as MC1R, located on the 16th chromosome. Her mother has red hair, and her father's father had red hair as well. This means that her father is a carrier, big R, little r, and her mother has red hair, little r, little r. The combination of these alleles gives a 50% chance that their offspring would have red hair. Both Erin and her sister have red hair. This is but one of the many examples showing that genes are inherited by the parents. So you may ask yourself, what is DNA? These molecules take the shape of a twisted ladder called the double helix. The structure on the outer edges are made up of alternating patterns of phosphate groups and 5-carbon sugars, known as deoxyribose. Nitrogen bases including thymine, adenine, cytosine, and guanine are attached to the sugar. The bases on one strand of DNA pair together with complementary bases on the opposite strand to form the rungs of the ladder. If not paired correctly, mutations can occur. Together, they control all the cell's activity and also transmit hereditary information. DNA is copied and passed to one generation to another. In eukaryotic organisms, DNA is found in a special place in the cell called the nucleus. Because the cell is very small, the DNA molecules must be tightly packaged, and this is called a chromosome. Besides the DNA located in the nucleus, humans and other complex organisms also have a small amount of DNA in their mitochondria. Mitochondria are small, membrane-bound organelles that are considered to be the power-generating station of the cell. Organisms inherit their mitochondria DNA, also known as mtDNA, from their mother. However, fathers cannot pass on their mtDNA because the male sperm has its own mitochondria, and the male mitochondria withers and dies when they fertilize an egg. We will be focusing on the importance of genetic genealogy, which is the study of one's ancestry. Because mitochondrial DNA only comes from the mother, there is only a slight change from generation to generation, but mutations do occur, but less frequently. Therefore, a person's mtDNA is almost identical to their maternal ancestors, even a dozen generations ago. In humans, the mtDNA is in the form of a circle, and they contain almost 16,500 nucleotide base pairs. Before humans began to travel the world, rare changes occurred in mtDNA, resulting in unique types on every continent. Therefore, most present-day mtDNA can be assigned a continent of origins. Geneticists do this by studying the HVRL region in the mtDNA. The HVRL is roughly 400 base pairs in length, and this is the region for the DNA molecule where mutations are most likely to occur. One type of mutation called a single nucleotide polymorphism, SNP, is a single base pair in DNA sequence that has been swapped out for a different nucleotide. For example, two sequenced DNA fragments from different individuals, AGA to AGG, contain a difference in a single nucleotide. By looking at the patterns of SNP in mitochondrial DNA, scientists can trace maternal ancestry back to hundreds of thousands of years ago. Scientists do this by comparing the patterns in mitochondrial DNA between different groups of people. If two people have similar patterns of SNPs in their DNA, they're most likely related in maternal lineage. Mitochondrial DNA helps scientists gather clues about ancient human migration patterns by studying the patterns of SNPs in modern-day population. When a group of people share similar SNPs, they are part of the same haplogroup. In conclusion, determining one's maternal ancestry is dependent on studying the mitochondrial DNA, or mtDNA. In our example that we used earlier, Erin, although she inherited her red hair from both her parents, her mitochondrial DNA, which determines her ancestry, only comes from her mother. This type of DNA is passed down from the mother and rarely mutates or becomes different than the person's maternal lineage generations ago. To do this, scientists examine the HVRL region of the mtDNA, where mutations are most likely to occur. Mutations are unique to the continent of the origin and therefore can be traced back to your ethnicity. Most mutations or SNPs, or single nucleotide polymorphisms, can help scientists trace back pretty far, even thousands of years ago. Thanks for watching, and please click on the link below to take our quiz on our project. We hope you enjoyed our video as much as we enjoyed making it.
I'm back.
I'm just gonna